Welcome to episode 16 of The Pulse of Physiology. I'm your host, Dr. Jim Davis, and today we're exploring the intricate world of G proteins and their critical role in cellular communication. G proteins are at the heart of many signaling pathways, allowing cells to respond to external stimuli with precision and efficiency. In our next episode, we'll delve deeper into neurotransmitters, but for now, let's focus on G proteins and how they work. So G proteins, or guanine nucleotide binding proteins, are molecular switches that play a key role in transmitting signals from the outside of a cell to its interior. They're often linked to metabotropic receptors, which are slower acting compared to ionotropic receptors, but allow for signal amplification and more complex responses. Here's how they function. They have an inactive state and an active state, and the G protein is inactive when a GDP molecule is attached to its alpha subunit. When it becomes active when the GDP is swapped for GTP, and this ultimately triggers downstream signaling events. Now, G proteins are composed of three subunits. An alpha subunit, which binds GDP or GTP, and is ultimately responsible for activating these downstream proteins. There's a beta subunit, which works alongside the gamma subunit to help regulate the G protein's activity. And like I just mentioned, there's a gamma subunit, which partners with the beta subunit to stabilize the whole complex. So how do they ultimately work? Well, the first step is signal reception. So a hormone or neurotransmitter binds to a G protein coupled receptor, often abbreviated as GPCR, on the cell membrane. The receptor activates the G protein, which causes GDP to be replaced with GTP on the alpha subunit. It's important to note that it's swapped, it's not converted into. Next, we have subunit dissociation. The alpha subunit, which now has a bound GTP, separates from beta and gamma. The signal is propagated when the GTP and alpha subunit binds to a target protein, and ultimately one of two things will happen. An ion channel will either be open or a second messenger system will be activated. And there are many examples of second messengers, and we will talk specifically about CAMP, DAG, and IP3. After this signaling pathway has happened, we need to inactivate the G protein, and so the GTP on the alpha subunit is hydrolyzed back to GDP. So in other words, we use water and we get rid of one of those phosphate groups. Ultimately, this inactivates the G protein and resets it for the next signal. Now, when it comes to the G protein pathways, there are several pathways, but we're going to dive into three major pathways involving G proteins. The first one is the G alpha S, and the S stands for stimulatory pathway. So it's activated when a hormone binds to the receptor, activating the G alpha S subunit. In this case, the GTP and alpha subunit bind to something called adenylate cyclase. And adenylate cyclase catalyzes the conversion of ATP into cyclic AMP, or CAMP. CAMP is our second messenger, and it acts as a second messenger by activating protein kinase A, abbreviated PKA, which regulates various cellular processes. And we'll talk about some of those later on in the semester. Ultimately, this pathway amplifies the signal, leading to effects like increased heart rate or increased energy mobilization. Then we have the G alpha I, or inhibitory pathway. It's activated similarly to the G alpha S pathway, but in this case, the G alpha I subunit inhibits adenylate cyclase instead of activating it. This ultimately decreases the production of CAMP because we don't have adenylate cyclase to convert ATP into CAMP. And as a result, if there's less CAMP, there's also going to be reduced protein kinase A activity. The G alpha I pathway can also lead to the activation of something called phosphodiesterase, which is abbreviated PDE. Now, PDE breaks down CAMP into AMP, which reduces the levels of CAMP in the cell. This dual mechanism ensures tight regulation of cyclic AMP, or CAMP, and prevents overactivation of downstream pathways. And we'll see PDE in a very similar role when we talk about vision. Ultimately, the G alpha I pathway dampens cellular responses and leads to reduced heart rate or reduced energy consumption. Finally, we have the G alpha Q pathway. Okay, and I don't have a good I don't know why it's called Q. I've looked it up before and haven't gotten a good answer, so I just refer to it as Quasimodo. Now, this G-alpha Q subunit is activated when a hormone binds to a receptor. Ultimately, GDP, it leaves, GTP comes in. GTP and alpha leave, and they activate something called phospholipase C, abbreviated PLC. PLC breaks down PIP2 into 2 second messengers, DAG and IP3. DAG activates protein kinase C, which regulates cellular function like contraction and secretion. IP3, on the other hand, triggers the release of calcium ions from intracellular stores, which amplifies the signal. Ultimately, G proteins are found in a variety of physiological processes, including regulating heart rate and cardiac muscle, detecting light in the retina, so that's phototransduction, sensing odors in the olfactory system, so that's smell, modulating blood vessel tone and smooth muscle contraction, and mediating hormonal responses in various tissues. So let's summarize with our key takeaways, and we've got 4 of them for this episode. G proteins are essential molecule switches, essential molecular switches in cellular communication. They work by activating second messenger systems or opening ion channels. Major pathways include G-alpha S, which is stimulatory, G-alpha I, which is inhibitory and can work with PDE involvement, and G-alpha Q, which has the second messengers of DAG and IP3. G proteins are found in numerous tissues and are critical for processes ranging from sensory perception to cardiovascular regulation. So that's it for episode 16 of Pulse of Physiology. I hope this dive into G proteins has clarified their role in cellular signaling. Please join me next time for episode 17, where we'll discuss neurotransmitters and their role in synaptic communication. Until then, keep questioning, keep exploring, and stay curious.